Hello, my name is Tristan,
And I'm Michael.
and our project is on the effects of sugary drinks on human teeth. So for our research method, we conducted experimental research. We tested the different samples of teeth we had, and we submerged it under a variety of sugary drinks. Our experiment lasted for 30 days, and after the 30 days are over, we completed the final analysis and recorded our results. The purpose of this project was to see which everyday sugary drinks would cause the most damage to our teeth. Our hypothesis was that if a tooth will be in constant exposure with the sugary drinks, then there will be damage present on the tooth. The severity of this damage will depend on the amount of sugar present in the drink and how long the tooth is submerged under the sugary drink.
To conduct our experiment, we have listed the materials we used over there, and we used real human teeth to conduct our experiment, and we also used small plastic containers to store them in, and we also had a variety of different sugary drinks such as Pepsi, Brisk, pineapple juice, apple juice, milk, 7-Up, and coffee. And then we used dental tweezers to get the teeth out and analyze them. And we also used syringe, tape, a pen, a phone camera to take the pictures. We used sugar and water to create some coffee, and we used a spoon. And for the procedure of our experiment, we first collected all the materials we needed, and then we labeled each container with the name of the drink we were going to put in it, and also the date, so that we maintained a record of what we have put in the container. And then after that, we took a picture of each tooth so that we know the condition of each tooth before we conducted the experiment. And then we put 22 milliliters of each drink into the container. We put the teeth inside as well and stored them at room temperature. And after 15 days, we came back, we analyzed each tooth, and we took a picture, as you can see here, and we recorded our observations. Afterwards, we put the teeth back in the containers and came back another 15 days for a total of 30 days from where we started to do a final analysis of each tooth and take pictures again. And what we saw was that there were no new cavities formed from the beginning. However, we saw some sugary coating on the teeth, which we'll explain later on in the presentation. And on this graph, in this graph, we have displayed the amount of sugar that's present in each tooth. So our graph is how much grams of sugar there is per 500 milliliters of each drink.
Since our project is mainly about teeth, it's only right that we explain the anatomy of the tooth. So the tooth is comprised of 4 main parts: the pulp, dentin, enamel, and cementum. The pulp is the inner part of the tooth, and it contains connective tissues, blood vessels, and nerves. The pulp has 2 parts, the pulp chamber and the root canal. The connective tissues and the blood vessels enter through the root canal and through the pulp chamber. Dentin is a hard yellow substance, and it makes up most of the tooth's area. Enamel is the hardest tissue in the body, and it allows us to withstand the pressure of chewing, and it protects the teeth from harmful bacteria. The thing with enamel is that once it's gone, it cannot regenerate because it does not contain any living cells, so it cannot regenerate like our bones, for example. And then finally, cementum. It's that over there. It's under the gum line.
And also, enamel is 95% minerals, 4% water, and 1% protein. And now we're gonna explain how cavities are formed. So in our experiment, we had the sugar and the teeth, which like I said earlier, did not form the cavities. And through our research, we realized that we were missing a key component, which is bacteria. So, like I said, through our research, we realized that we needed sugar, the tooth, and bacteria in order to form cavities. So after we drink sugary drinks, especially ones that contain sucrose, there's sticky glycoproteins in the sucrose which adhere to our tooth. And then once in our mouths, the sucrose, which is a disaccharide, hydrolyzes into glucose and fructose. And the fructose is what the many bacteria in our mouths use, such as the Streptococcus mutans, which is one type of bacteria that has been studied the most to cause dental cavities. So the bacteria then use the fructose in a process called glycolysis, which results in the formation of lactic acid. This lactic acid then increases the acidity of our mouths and decreases the pH. This then results in the dissolving of the calcium phosphate in our enamel. And like Tristan said, the enamel doesn't grow back. So once the enamel is worn down, the acid and the bacteria continue their way through towards the middle, towards the pulp cavity, where all the nerves are, and that's the pain we feel when we have cavities.
So in conclusion to our project, in the beginning we expected cavities to be formed on the tooth, but through our research we learned that we were missing one main component, which was the bacteria needed to cause the cavity. As you can see from the picture over there, we need bacteria and sugar and time to cause a tooth cavity to develop. Regardless of the bacteria, we still saw the sugar present on the tooth, which was the sticky glycoproteins that Michael mentioned. We also learned that the lactic acid, which lowered down the pH and would wear the enamel down even more. So overall, what we learned was that it's really important to maintain oral hygiene and to always brush, floss, and use mouth rinse to prevent any further damage from happening. Because from these pictures over here, this is only the beginning of dental cavities, and if bacteria were to be present in this, then severe damage would be caused.
That's it.